Welcome to the Department of Medicine CMC Vellore. This is our academic breakfast. Though it's night time in San Diego, and Suresh must have just finished dinner, I hope. So today's talk is going to be on drug discovery. So just few brief words about Dr. Stephen Antonisamy. He's my friend, my brother. He received his training in MSc in physics in the Indian Institute of Technology in Kanpur. I think one of the first few people from Vellore to have got into IIT, a genius from that age. Subsequently, he went on to the Indian Institute of Science in Bangalore, then did his PhD in molecular biophysics. And after that, his postdoctoral fellowship in structural based drug discovery. And he did it in trypanosomal diseases in the University of Washington in Seattle. Then he joined the SJX Pharmaceutical, and currently he's working with Eli Lilly on drug discovery. He does crystallography and stuff of the dreams of most of us. And today he's going to share some of those dreams with us. Today's talk, you may wonder why medicine department needs to hear about drug discovery. But I think in the era of COVID, we all are looking for new drugs and looking for some magic cure for something. So he's going to take us through the process of how a drug is discovered and how one decides whether it may work or may not work. So today's talk will describe the current drug discovery process and the rationale for a target-based drug discovery. And he will also talk about small molecules and antibody drug discovery. So it's really an apt talk for us at this time. And I really wish more students come and join and listen to this talk. After this talk, Suresh will stay back, I hope, for a few minutes of Q&A. Please keep your questions on the chat board. And after the talk, Mohan will take Dr. Suresh through the talk, to the questions which are there, if there are any. And after that, we will put a recording of this on the YouTube. So if somebody missed this talk, you can always share the link with them from our department YouTube channel. So once again, a warm welcome to Dr. Stephen Antonisamy. We're very honored that you accepted our invitation. So on behalf of our department and CMC, thank you, Suresh, and over to you.
Thanks, Tam. Thank you for the most generous introduction. I'll switch off video while I'm talking so we can focus on the slides. Thank you also for Dr. Mohan for arranging the logistics. I really appreciate Dr. Thambu David inviting me to speak to you today. As you may have guessed from the title, my talk is about drug discovery, and I will share with you an overview of the process employed by pharmaceutical companies to discover new medicines. I was not sure exactly at what level to place the talk. I hope I hit the right level because I think the audience might be of different experiences in medicine and in research. We will touch on several topics since modern drug discovery is a multidisciplinary endeavor. Gone are the days when you had a single lone genius scientist sitting in a lab with a couple of assistants discovering new medicines. Modern drugs discovery is a team sport with a large team composed of cross-functional team members, more often than not located in different places in the country or even in different countries. In the first half of the talk, we will examine the whole drug discovery process, how drugs work, and how do we go about discovering new drugs. And then for the second part of the talk, we will get into some detail of what is called structure-based drug discovery, which actually is my area of specialization, as Thambu mentioned. Okay, so as all of you know, human beings have been doing some version of drug discovery for research for many millennia. Ancient cultures like the Indian, Chinese, Egyptian, and Native Americans have what you may call healing traditions, where they use herbs, roots, minerals, or animal parts to treat human disease. Now, their approach actually has similarities to what we now call clinical trials in modern drug discovery. In the sense, they experimented with different herbs and natural materials and found some which had beneficial effects. And then this knowledge was passed down generations and sometimes even documented in writing. Oftentimes, these medicines prepared from natural sources were mixtures of complex molecules that plants synthesize. And when we ingest them, these medicines work on the body in multiple different ways to produce the desired effect. Now, many of these remedies have now been analyzed, and for some of them, we actually know how they work. We know what the active ingredient in that cocktail is and how that interacts with the body. Classic example is aspirin, or by its scientific name, acetyl salicylic acid. Now, more than 2000 years ago, people were using extracts from the leaves and bark of the willow tree to treat pain, fever, inflammation. But it was only when scientists discovered what the active ingredients was, and then most importantly, found a way to synthesize that active ingredient, acetyl salicylic acid, in the laboratory, that we could produce it in a sufficiently large scale, and then aspirin was widely adopted as a medication for pain, fever, and inflammation. Now, the really interesting aspect, as you know, is that aspirin continues to find a use in modern medical practice. But now we have a much better understanding of how it works. And we also have the tools to try and find a better aspirin if we want to improve it and decrease some of the side effects like gastric effects that aspirin has. Now, the last few decades have been really seen breathtaking advances in molecular biology, in genomics, proteomics, and structural genomics. And every year we are discovering new insights into how our bodies work and how molecules come together to make life happen. It's also true that with each discovery that we make, we also learn how much more there is that we still don't understand. There are many diseases for which we don't have good treatments, and even good medicines that once worked sometimes may no longer work now. This loss of efficacy could happen due to very different reasons. It is a constant battle, an arms race, so to say, between the scientists, doctors, and patients on one side, and infectious bacteria, virus, and cancer cells on the other side. The truth really is that a number of us on this call are going to get cancer in our lifetimes. And for some of those cancers, it's possible we have the treatments now, but there are others for which we don't yet have treatments. The other scary disease that some of us will get is dementia, or brain disorder. As human lifespans have increased, many of us have friends and family members who are affected by Alzheimer's disease. And right now, we don't have any cure which has a significant effect on Alzheimer's disease. Still, the revolutions in molecular biology, biotechnology, proteomics, and genomics have provided us with an understanding at a molecular level about many of these diseases, and also possible methods we can use to treat or cure these different diseases. And if there is one message I would like you to remember after today's talk, it is the idea that drug discovery today is based on an understanding of the molecular basis of disease. That today's medicines are designed after understanding how diseases happen at the molecular level. It involves the identification of a protein, what we call a drug target, whose overactivity or underactivity causes a disease. And then we try to find a drug that will modulate the activity of this target protein to treat the disease. So in this talk, we are not going to cover other modalities such as gene therapy or vaccines or things. We are only going to talk about target-based drug discovery, which predominantly is the way that most molecular drugs, either small molecule or large molecule. When I say large molecule, I mean an antibody drug. When I say small molecule, I mean a molecule with 10 or 20 atoms, how they work. This slide might be a little basic for this audience, but so I'll quickly skim through it. To understand how drugs work, like I said, we need to understand it at the molecular level. Now, if you had a microscope big enough, you'll see that everything in the world, both inanimate objects and living things, are made of atoms. Of course, these atoms don't exist in isolation, but they form covalent bonds to form molecules. In drug discovery, if a drug molecule is made of a small number of atoms, 10 or 20, we call it a small molecule drug. But then there's another type of molecule that is present in living systems. These are called proteins. Now, protein molecules are made of thousands of atoms. What you see here in the middle is a protein molecule, where I'm showing every atom of the molecule. It looks kind of crowded and confusing, but this is just one representation. There are other ways of representing protein molecules which we will look at, which is a little bit easier to consume for the human brain. Now, as you know, a multimolecular organism like a human being is made of trillions of cells. Now, each cell is like a factory. And within that factory of a cell, you have proteins, and the proteins are the machines in the factory. These proteins in the cell are constantly making molecules, breaking down molecules, converting molecules, reading DNA code, synthesis. And it's truly amazing what goes on in each and every cell of ours. Now we know that cells are grouped together to form organs, and the organs make up the whole body. But pretty much all the action happens inside each of the cells. And it is the proteins which make everything happen. Your body is made of trillions of cells. Each of the cells is like a factory, and they all have to work together for you to function. Your body is constantly replacing the old cells with new cells at the rate of millions per second. And the life cycle of each cell is carefully controlled, and it is controlled by the proteins within each cell. Now, we are all familiar with this central dogma in biology. The DNA contains the sequence of the protein encoded in 3-letter codes. And for each of the proteins that the cells make, this information is transferred to the messenger RNA. And then the protein RNA complex, which we call the ribosome within the cell, reads this DNA code and synthesizes the protein. But then is where the magic happens. Freshly synthesized protein is like a long noodle of amino acids. These amino acids are the building blocks of protein. They are like beads on a string. Then depending on the sequence of the amino acids, the protein will fold to form a unique structure. Almost like magic. Before dividing, each cell has to make a copy of the entire DNA. Imagine while making this copy, it makes a mistake. Now you have a DNA in one of these two cells which divided, which is slightly different in its sequence. It has a mutation. Now the protein which this piece of DNA codes will be a little bit different. Now imagine the job of this protein was to trigger cell death. Maybe it was involved in triggering cell death of the cell. Now, when the lifespan of the cell is over, this protein was supposed to trigger the death of that cell. Now this protein, because of this mutation, is no longer able to do its job. Now the cell will not die when it's supposed to. It becomes an immortal cell, a cancer cell. And soon you will have a tumor when this cancer cell divides, multiplies, and you will have a tumor growing on you. I mean, in fact, as sad as it is that many of us will get cancer, it is even more surprising that more of us don't get cancer. I mean, the cells are dividing millions of times every second. And the fact that the cells make mistakes is natural. But the body has many defense mechanisms where it identifies these mistakes and corrects them. And that's the reason more of us don't get cancer, because many of the mutations, those cells are targeted, the proteins are killed, the cells are killed and taken away. But you can imagine when the protective mechanism fails, you can easily make a mistake in this DNA duplication when cells divide, and then you have cancer. Now, like I said, proteins are the machines in the cell which do all the work. Proteins are macromolecules. Like I said, they're made of thousands of amino acids. Here, each amino acid is colored in a different color. This basic building blocks of proteins, there are only 20 of them in the human body, 20 different amino acids, and they can occur at different number of times. Then depending on the order and the sequence of these amino acids, this protein is going to fold to form a unique structure. Now, why do I keep harping on structure? Why is the structure so important? The structure of the protein determines its function. Like I said, the protein is like a machine. The shape of this protein is going to determine how it is going to do its function, how it is going to interact with other molecules. When I'm talking about a protein interacting with other molecules, I'm talking about interactions between one protein molecule and other protein molecules, interaction of the protein with small molecules, interaction of the protein with the substrate small molecules. It reacts with the chemical reactions it catalyzes. Pretty much for the machine, for the protein to work, it has to interact with other molecules, and it does that by the shape of the protein. Now, this is a protein structure which was solved by my friend Madhusudan at the University of California in San Diego in the lab of Susan Taylor. It's a very interesting structure, and we'll come back to this family of proteins again a little later in the talk. So as I mentioned, most drugs work by interacting with protein molecules in the cells of your body. The vast majority, pretty much invariably, all the drugs that you take go into the body, I mean, depending on how they are delivered, and then they interact with the protein molecule. So if you want to design a new drug, you need to know the shape of the protein to do it more efficiently, more quickly, and try to get the best drug molecule which will bind to your protein molecule. And that is the reason why the structure of the protein is important. It's not only important to understand the function of the protein, but also in drug discovery, when you're trying to design a small molecule, it becomes very critical. Now, to recap, what have we talked about? The modern drug discovery process involves identifying a protein, what we call a target, which is implicated in a disease process. And then what we do is we oftentimes develop assays as some kind of way to screen compounds or small molecules which will interact with this protein target. And once you have done that, we try to optimize this molecule. The initial molecule you identify might not be very good in, say, binding to your protein. Then potentially using the protein structure, we synthesize other molecules which are slightly different from the initial molecule we identified so that it can bind even tighter to your protein. And then we have to look at properties like absorption into the body, distribution, how it gets metabolized. We want the metabolism to be there so it doesn't stay in your body too long, but we don't want it to be too much that it's kicked out before it can even have its effect after that excretion. And then we want to talk about toxic side effects. So anytime you have a protein you're targeting and you have a potential drug molecule which is interacting with a target protein, if it interacts with some other target, some other protein out of the 20,000 proteins you have in your body, you're going to have a side effect. Because each protein is doing a function and you're trying to interact with one protein molecule which is causing your disease. And if your small molecule goes and binds to some other protein molecule which is not involved to the disease, you're going to have a side effect. Now, hit, lead, and candidate are just terms used in the pharmaceutical industry to call the initial identification of a molecule which has some activity against your target, and then you improve it to a lead molecule which has slightly better activity. And a candidate is like a molecule which you have optimized, changed its shape or its actual structure so that it now binds and hits your target so hard that you can now start testing it in human beings. So you take it to clinical trials, and in US, you have a FDA approval, and then you market it. Okay, this is a slide about drug delivery, which possibly this audience is more familiar with than I am. But irrespective of how you deliver your drug, unless it's a topical drug, if it's a systemic drug, you deliver either orally, where it goes into your intestines through your intestinal wall and gets into your blood, or inject directly into your blood. Now, this drug is going to go all over your body. I mean, maybe you have a tumor in a certain place, but this drug is going to go all over your body. And that is kind of a problem, right? I mean, so you have to make sure the drug stays in your body sufficiently long enough to have its effect. You have to make sure that it doesn't affect normal tissues and things. It affects only the parts of the body in which you have actual disease. And then you have to make sure it only interacts with the protein molecule you want to interact with. Whereas, like I said, there is so many other protein molecules in your body that this drug can go and bind and cause all kinds of side effects. So that is the true challenge of drug discovery. The average human body, like I said, is composed of around 37 trillion cells. And each of those cells have about 100 billion protein molecules. And there are 20,000 different protein molecules in this human body. We are trying to find a drug that specifically binds to one specific protein molecule out of the 20,000 protein molecules for it to have the desired medicinal effect without binding to the other 19,999 protein molecules, which could cause side effects. Now, let us look at this problem from the other side. Let's look at the spectrum of small molecules that we know exist in nature. I mean, there are about 130 million molecules that are registered in the Chemical Abstract Service maintained by the American Chemical Society. Now, if you consider all the molecules that you don't exist yet, but you could potentially synthesize that will have drug-like properties, the so-called chemical space of potential pharmacologically active molecules, that number is 10 to the power of 60. I mean, a mind-boggling number. And a drug discoverer's job is to find out which of those molecules he should synthesize that will modulate the target and without hitting the other 20,000 protein molecules in the human body. It's like finding a needle in a haystack. Now, coming back to this family of proteins called protein kinases, which is kind of an example of how challenging things can get. Now, protein kinases is a family of proteins. They all do the same function. They catalyze the same reaction, have similar sequences and structures. They act on different substrates. So a protein kinase, what it does is it takes a phosphate group from ATP and transfers it to the side chain of another protein. This reaction is called phosphorylation, transferring a phosphate group from ATP to another protein. Now, this addition of a phosphate group to this other substrate protein is like a switch. It may activate that other protein or it may deactivate the other protein to control its function. Now, this is a switch that, like I said, there are 500 different protein kinases in humans, and each of them gets switched on by one and the other. So one protein kinase will switch on another protein kinase, which will switch on another protein, and they all have very similar structures. They all catalyze the same reaction, but there are subtle differences between them. And you are trying to inhibit one of those protein kinases, which is involved in a particular cancer mutation, which you have identified in patients. And now you saw the structure of that protein kinase. And then you saw the structure of the protein kinase with the mutation which causes cancer. And you're trying to find out what is a small molecule which will bind to the mutant protein, not bind to the native protein, not bind to any of the other 20,000 proteins you have in the human body. You can imagine how challenging that's going to be. Now, the properties of that small molecule has to be such that it gets into the body, it stays the amount of time you want. So you have this safety window, right? Below, if the concentration it gets to in the body and the time it stays, the area under the curve, what they call concentration versus time, if that is too low, you're not going to have any efficacy. If it is too high, then it starts, even a good medicine becomes a toxin. Okay, so this next 3 slides are just snapshots of different pathways. Like I said, we have around 500 protein kinases, and that is the way the machinery in the cells, the proteins talk to each other. One will add a phosphate group to another one, which will add a phosphate group to the third one, and then the feedback mechanisms. So whenever a cancer is caused by a particular mutation in a protein kinase, you will realize that the effect actually goes down in waves to other protein kinases which are interacting with this particular protein kinase. And as you can see, there are feedback mechanisms, the pathway divides, and these things. See, CDK is another family of subfamily among the protein kinases which is involved in cell division. And you can see there's so many feedback mechanisms. So when you think of inhibiting a protein kinase, you never know what other effect it can have by inhibiting it. Sometimes the feedback mechanism causes more of the protein kinase to be produced when the body realizes, hey, this protein kinase level is being inhibited. So I'm showing you just these 3 slides to just show the complexity of the multiple things that go within each of our cells to keep it functioning in harmony with all the machines, the proteins interacting with each other, talking to each other, catalyzing things, and how difficult it can get to identify one protein which causes a cancer, but then to go and target that protein to inhibit it. So this is again kind of a pause point where we look at the whole drug discovery process. So the initial step here is the target identification, where we identify the gene or the protein which causes a particular disease or is implicated in a particular disease. And then we try to see, even though we have seen it in patients, is inhibiting or increasing the activity with the agonist as opposed to antagonist of that protein target, would that cure the disease? So we can do target validation experiments in cells. Say we have the cancer cells, you grow the tumor cells, see if you can inhibit this protein, particular protein, and then does it have the effect you want? And then we talked about hit, lead, and then candidate. So after that, you have this small molecule which interacts with the protein molecule, and then you try to test it out in different cells. You do the biochemical assay. If you can kill the activity of the protein kinase, prevent it from doing the phosphorylation, adding the phosphate from ATP to another one, and then try to improve it. And then you test it in animals, maybe rats, dogs, maybe primates, and then it's ready for clinical trials when you test it in humans. So as much as you can do in vitro in cells and then even in animals, you never know till it enters the clinic what other complicated things that exist in humans which don't exist in primates or other lower animals. But if after testing in sufficient number of humans, you find it is efficacious, then you apply for approval and then it's approved and then you can test the drug. Now we're going to take a pause and talk a little bit about large molecule drugs. Now this is just a overview slide of innate and adaptive immunity. I'm sure this audience is very familiar, possibly more familiar than I am, about the immune system. But we're going to talk about one specific component of the immune system, which is the antibodies, which are molecules, large molecules, protein molecules produced by our body to fight against anything which is foreign. So these are like the guardians of our body. These are antibody molecules. I'm showing you a sample of antibody molecule, which again was solved in San Diego in the lab of Erika Sapphire at the Scripps Research Institute in San Diego. Now, scientists said, okay, we have this protein target. We are trying to inhibit its effect. We are finding small molecules which can go and bind into a pocket into this target. But this body, our human body makes these antibodies. Can we use antibodies to make medicines? Basically, instead of the body making antibodies to hit against foreign things, can we design and synthesize antibodies in the laboratory which will go and attack a particular target? So antibody drug therapy has become a big part of modern drug discovery. In fact, most pharma companies do half small molecule drug discovery, where you have small molecules with 10 to 20 atoms, and then large molecule drug discovery, where you have antibodies against different targets. So there are different ways, therapeutic uses for antibodies. The most simplest way is you find target protein similar to what you would do with a small molecule. Instead of inhibiting with a small molecule, you use an antibody, which you can design antibodies with exquisite specificity, which will go and bind to a target molecule and prevent its action. Now, you can design antibodies which go and target to cancer cells, and you can attach a small molecule to that, which when the antibody sticks onto a cancer cell, the small molecule will now cause the death of that cancer cell. So you can do targeted cancer therapy by antibody drug conjugates, ADCs, antibody drug conjugates, where you have had a drug which you conjugate or bind to an antibody and let the antibody go and find cancer cells and go stick to them. And then one of the challenging things with small molecule drug discovery is when two proteins interact to cause a disease, something in the interaction is not quite right, cause a disease, and you want to disrupt it, it's very, very difficult to do it with a small molecule. But with the antibody, it becomes a lot easier. Like I said, antibodies are exquisite specificity, and by their large size, they can interact with a large protein surface, and then they can help you deal with that disease. And then you can tag cancer cells for the immune system. Okay. There is something really acutely distressing and kind of heart-rending about autoimmune diseases. I mean, antibodies, your immune system is supposed to protect your body. It's almost as if you hired a security guard to take care of your grandmother's house, and this dude breaks into your grandma's house and steals all the jewelry. I mean, it is so tragic when you think about autoimmune diseases. It is like your own body's immune system no longer recognizes what is foreign, what is yours, and start attacking your own body. But then again, once you have identified which part of the immune system is being enhanced, then you can select drug targets involved in the immune system and then target them to see either with small molecules or with large molecules whether you can hit. And there are many approved drugs which seem quite magical in treating some of these diseases. So this is kind of a pause point. I don't know whether there are any questions we should take at this time. Thumbs, what do you say, or shall we wait for questions at the end?
We'll take questions at the end, Suresh. That's usually how we do, so
Perfect.
that so just to get the flow
Okay.
going. Okay.
Sounds great. So
Okay.
we're going to shift gears a little bit. But by now, I hope you have understood that protein structure is so important to its function, and also that if you're going to design a drug, it will help you a lot. Especially if you're designing a small molecule drug, it will help you a lot to understand the shape of the protein, so you can design a small molecule which will bind to the pocket in the protein and prevents its action. Or in some cases, you want to increase the action. You have a tumor suppressor gene which is now not working anymore, and you want to enhance that tumor suppressor gene. And then you still need to know the shape of the molecule, so you can design your small molecule drug which can go bind to it. So let's take a closer look at what protein structures give us, what it means, and how we go about determining protein structures. Now, today there are two predominant ways of determining protein structures. Crystallography is a classic method. It's been there for like decades, and it's become such a high throughput, highly efficient way of determining protein structures. The one caveat with crystallography is before you can determine the structure of a protein, you need to grow these crystals of these proteins. So you take a protein in solution and add something what you call a precipitant. Actually, it's a crystalline, something to decrease the solubility of the protein. And then when it falls out of solution, it forms a crystal, almost like a sugar crystal or a salt crystal, except this is more soft like cheese crystal. I mean, this is soft like cheese, but it's actually a crystal in the sense it has long range order. When you hit this with X-rays, you get spots where the electrons in each of the atoms in your protein crystals this contains like tens of thousands of protein molecules. Each of those diffract these X-rays and you get spots. Now in the computer, you can do a mathematical Fourier transform and get the structure of your protein. So that is crystallography. Now electron microscopy has been used for decades. I mean, it's another technique which is used for decades. It was never really used for solving high resolution structures because most of the maps you got from uh, most of the structures you got were very low resolution. You couldn't see individual amino acids. You couldn't see individual atoms for sure. In X-ray crystallography, you can see position of each atom in your protein molecule. Here, you couldn't see such things. But then what happened was 7 years ago, uh, there was a major technological advance in electron microscopy. They designed a new detector which could detect a single electron. Now, once you had this detector, it was coupled with advances in the microscope hardware itself. In parallel, people design more powerful computers and computer programs which can analyze this data. And today, we are in the middle of what has been called the resolution revolution in electron microscopy. In fact, a couple of years, three of the scientists involved in developing cryo-electron microscopy were awarded Nobel Prizes just because of the advances which have happened here. Today, you can determine cryo-electron microscopy structures through cryo-electron microscopy, which are of similar resolution as what you could get with crystallography. And unlike crystallography, where you have to go grow crystals of the protein, which sometimes can be very challenging, here you just take your protein in solution, flash freeze it, hit it with electrons, and then collect enough data because each of these protein molecules, the image of them will be very noisy. But you collect enough. Like I said, the detectors are much more powerful. The computers are much more powerful. You can collect a lot of images and then do computer analysis to get the structure. So these are the two predominant methods right now to determine the structures of proteins. Let me take a quick look at the time. I think we're doing okay. Okay, so of what use is protein structures? I think by now you would have already realized how useful a structure can be. So when the initial structures were discovered in the 1960s of hemoglobin and myoglobin, they were kind of a novelty. They helped you a little bit understand the function. Nobel Prizes were awarded for it. But then people really did not know how to use the structure to find new drugs. But then they used it to understand the biological function because, like I said, the way protein interacts with other proteins and things is based on its shape. Like I said, last few decades, we have had big advances in computation and biotechnology and recombinant proteins where we can actually get E. coli or baculovir insect cells or yeast to make our proteins. And then the speed at which we can determine structures. Now, instead of collecting in a small X-ray generator in your laboratory, you go to a synchrotron, which is a government facility usually, or a facility by the science department of a country, where you can collect data within minutes and then have a structure within hours. So this ability to rapidly get structures has dramatically changed how you can go about optimizing your initial hit against a target to improve its affinity to the protein and then finally developing it into a drug. So once you have a hit against a protein, you determine the structure of that small molecule with your protein, and then you see how you can change that protein to increase its affinity to how you can change the small molecule drug to increase its affinity towards your protein. And you do that quickly enough, then the chemist can use that information to generate, to design and synthesize the next set of molecules, which will bind even tighter to your protein. And then you do it a few cycles of that, and then you get your small molecule potential drug, which binds very tightly to your protein. And this goes through the whole process. When I first made this slide, I did not have the cryo-electron microscopy, but just based on the revolution in the, like I said, in the last few years, this has become an equivalent technique with X-ray crystallography. You grow your cells either in E. coli or insect cells or in yeast or sometimes in mammalian cells in a beaker. You express your protein, purify it, and then if you're doing crystallography, you crystallize it, or if you're doing cryo-electron microscopy, you make grids. The grids actually have holes, and then you apply your protein solution, and then you flash freeze these into liquid propane, which freezes it even faster than liquid nitrogen. And then you collect, if it's a grid for electron microscope, you collect it on an electron microscope. If it is crystal, you collect it at synchrotron, and then determine the protein structure, which will look the same whichever way you determine the structure. This is another view. We at the end of it, depending on time, I'll show you some other views of actually a drug molecule bound to a protein, and maybe that will illustrate how useful that can be. So when you have a structure of a potential drug molecule bound to your protein, you look at what are the interactions. I mean, there are different types of proteins, and ligand is basically your potential small molecule drug. It can form hydrogen bonds, it can form van der Waals interactions, and so-called non-classical interactions, which are interactions between a chlorine atom and a carbonyl oxygen. And then you can see, is your small molecule bound in a good energy conformation? If it is not, then that could possibly cause weak binding to your protein. So you can see if you can how you can change your small molecule to bind even tighter to your protein. And other aspects, we won't get into the details of that. But in essence, the major idea is that you are trying to change this potential drug small molecule, which binds to this protein so that it binds a little tighter. And then you're trying to make changes to this so that it behaves better. The pharmacokinetics in the body is better. It's absorbed better. It's not metabolized too much, metabolized just the right amount. If you're going to treat something in the brain, it crosses the blood-brain barrier. The different molecular properties of your drug, which you have to pay into attention in case you want it to be effective. And like I said, drug discovery is a team sport, cross-functional colleagues from all over. And within that larger team is a sub-team, what we call the design team, where you have the structural biologist, the crystallographer, electron microscopist who determines the structures, then works with the computational chemist who in the computer will try and see what molecules we can design. And then you talk to the chemist who's actually going to make that and say, you might design a very cool looking molecule in the computer, which the chemist will say, no, that doesn't exist. You can't design that molecule. It won't be stable. So this is a small sub team which is involved, the design team, which designs these new molecules which can potentially turn out to be drugs. And this slide just kind of goes into the detail. Just one structure of a protein is not enough, because you see your initial structure with a small molecule that you call a hit. It's not really efficient in inhibiting the protein. You want to improve it, and then you make some small changes. It's possible that the small molecule will just flip in the pocket of the protein, and now any change you make will have a very different effect on what you thought based on your initial structure. So at every stage, as you're trying to improve your small molecule and make it into a drug, you need to get the structure of the small molecule with your protein. And so in the bottom, you have this design hypothesis. You design your next cycle of compounds, the chemist synthesizes, you test it in your assays in cells, in animals, and then you determine the structures. And that will guide you in what you should make and what you should not make. We'll talk a little bit more about that when we look at the actual structures. So what are the limitations of structure-based drug design? Well, the structure you're looking at is a snapshot. Proteins are in solution, they move around, they take different conformations. You are taking just a snapshot of that. In the cell, of course, the protein is interacting with other proteins. It's all in a gooey mix of things. There are so many other things going on. So we should remember that even though our structure is reflecting what possibly exists in the cell, it is possible there are some significant difference, maybe involvement of different binding partners, other proteins, post-translational modifications. I'm pretty close to the end of the talk, so we have sufficient time to take questions. So this is just a snapshot, I think, from 2016 of different structures we had solved in structural genomics, which got, as Thumbs mentioned, got bought by Eli Lilly. And we have a lot more structures. Now, many of these we have deposited into the PDB so that others can use them to design new structures. I'll just take a minute to share something else. Stop sharing. I'm going to try and share structure of a protein. Okay, so this is the structure of a protein. Here what I've done is the small molecule is shown, all the atoms of the small molecules are shown. But of the protein, what I've done is each amino acid is connected to the next amino acid, and I'm just showing you how that long noodle of a protein exists. I mean, I can color this as from blue to red. So this is a structure of a protein kinase called c-Abl, which is bound to this drug called Gleevec, which you might be familiar with. It's a drug against CML, chronic myeloid leukemia. This is one way of looking at a structure. Another way of looking at a structure is looking at every bond of the protein, and then, but most often we look at it, the structure of the protein, as a surface. And here we have the bonds shown, but also the surface. And then you can see the small molecule bound here. And this shows you how the structure can be used to design a better molecule which will bind and inhibit your protein even better. You can see this empty space here. Maybe if you add another carbon atom, you can fill the space and bind even tighter to this protein. Or maybe if you add a nitrogen atom which will be positively charged, this red negatively charged atom from the protein will form a favorable interaction. So those are the things you think about when you're trying to use protein structure to design the next better molecule which could potentially be a drug. I have only one last slide, which I think is okay. So this is my last slide. It's just a plug for structural biology, either whether crystallography or cryo-electron microscopy. Drug discovery is hard enough. Don't try it blindfolded. Thanks, Thumbs. I'm happy to take questions now.
Thank you so much, Suresh. That was excellent. I think it was an eye-opener for many of us. We have some idea of how drugs are developed, but I think it's fascinating to see the actual process that a drug goes through. Maybe I'll just start off with a question. How do the first part of it? How do you know what to target? Like, who tells you that this is the place you must target against? Like, who tells the structural biologist that, or somebody like you, that this is the area where if you target, something will happen?
Correct, Thumbs. Good question. So multiple ways, Thumbs. So all the pharma companies have their own research units which do biology stuff to see how different kind of pathways within the cells work. Then there's publicly funded stuff, academic universities publish stuff. But to give a typical example, let's take cancer. Now, when tumors are there, now people quickly sequence the gene, right? Sequence what are the mutations in that. So for every cancer, CML, everything, then they know, hey, these are the genes which are mutated in this cancer. Oftentimes, like, it gets pretty complex now. Previously, people thought each tumor will have one mutation. But then the fact is that, a decade ago, people found out it is heterogeneous. The parts of the tumor have certain mutations. Because the cells are dividing so fast, others have certain mutations. But that is one primary way you can find out which is the protein to target based on sequencing a tumor, for example. Other ways are in diabetes, what is the pathway in which the feedback mechanism, insulin is recognized by which receptor, and then you see what happens. So you try to increase the sensitivity of the receptor by finding an agonist, which is something which will enhance the activity of that receptor. So there are multiple ways. Basically, you have to see. Finally, it comes down to each protein. Is it involved in a disease? And that is where the target validation is. Can you find out if I modulate, if I increase the activity or decrease the activity of this particular protein, am I going to cure a disease? And for cancer, it is a little bit more transparent, but then again, it becomes complicated. Does that help?
Yes, absolutely. I think there are some questions. Mohan, go ahead.
Sir, there is one question on comment. Amazing technology, sir. How do you determine the exact dose of a drug?
Yes, so that's a complicated thing. So, in terms of what is the exact dose of a drug, it has to be more sufficient for it to have efficacy, but not too much for a toxicity, right? So you have that window, and the larger you make that window, the more leeway you have to work with. And many times you have to test it in animals to see, I mean, what is the highest dose you can go before you start seeing toxicity? What is the lowest dose you can dose when you have efficacy? And again, I think in clinical trials, you have to still do it again to see in humans what is the maximum dose since we start seeing toxicity. So you can make theoretical predictions, but then finally you have to actually try it out and test it. That's why your phase one, phase two, phase three. Phase one is kind of a safety study. Then phase two is a large efficacy study, and phase three is the really large study where you say, okay, now before you can actually approve it, do a phase.
Sir, next question, sir.
Yeah.
What softwares do you use for drug design?
It depends on which part of drug design. So looking at protein structure is one software, but at every stage, like I said, it's a cross-functional, multidisciplinary effort. So each person, if we go back to, okay, if we go back to, so this is just one part of. This is just one part of this thing. I mean, this has a lot more granularity if you look into it. In lead generation, there are different things in lead profiling. But if your question is what software do I use to look at protein structures, there is a free software, it's called PyMol. So a lot of public structures are deposited into the public database, protein data bank, PDB. You can search for that PDB. And then if you want to look at a structure in the PDB, you can download this free software, very easy to use, called PyMol, PYMOL. Python molecule, PYMOL. So it depends on what part of it. I mean, we do a lot of modeling in the preclinical stage so that we don't have to test animals. We look at all the data of all the compounds we have, how they behave, and then predict how they will be absorbed, how they will be metabolized. Does that answer the question? So it depends on what you're talking about.
The request to all participants, you can drop in your questions in chat box, or you can unmute yourself and you can ask question directly to sir.
There's a question somebody's asked, does India have a synchrotron? I don't know if I'm
No,
pronouncing
India does
it
not
right.
have a synchrotron. We don't have a synchrotron in India. But the thing is, right now you can freeze your crystal and send it outside. So when I was doing my PhD, the synchrotrons were not that popular. They're just coming up. We used to send sometimes to Japan has one, Australia has one. So we used to send it to different places. Yeah, we don't have one in India, but right now in US, Eli Lilly has its own touch or one beam line at a synchrotron in Chicago, but when it's not working, when they have routine maintenance 3 times a year for a whole month, we ship our crystals to England or France. So you could potentially freeze your protein crystal and send it to a synchrotron in another country and collect. So US collects a lot for groups in other countries. I mean, our beamline actually for any academic group who wants to solve a structure, we will collect data for free.
What is the general time from the time like somebody has a disease, let us say even it's COVID, and then a virologist or somebody identifies a target till the clinical trial? Is there can you just take us to how much time each of these steps in the circle takes?
It depends on the therapeutic area, Tom. So if you're talking about cancer, sometimes it can be much faster because cancer is such a terrible disease that you are willing to take certain amount of toxicity. But if you're talking about say diabetes or something where a large number of people will take your drug, then your trial itself will be much longer. So in terms of that, so we are talking 10 years is kind of around the average. But by the time you've done your clinical trials, like that's 5 years or so, and then you've done your preclinical. The preclinical stuff we are trying to reduce to 2 years, right? So from the time you start identifying your target to when you have a candidate. But usually it's 3 or more years actually from the time you identify your target to actually have a candidate you can test it. So it's very variable. The COVID situation is amazing. So you can see that since it's such a devastating, I mean, it's transformed our world, right? So people have just sped it up by order of magnitude, by 10 times faster. I mean, even the vaccine things, they are doing it. I mean, of course, we won't have it before the US presidential election, but we might have it next year, which is like super fast for any kind of a vaccine trial, for any kind of a drug. And that's the same thing, right? I mean, the FDA, which is the approving agency in the US, is giving emergency use authorization for different kinds of drugs, which can say reduce the immune system, because part of the toxicity from COVID is that you have this immune flare-ups, right? So different drugs which have been approved for other things, they are giving emergency authorization. So it can vary between like 5 years to 10 years.
Okay.
So there is another question coming up. How much it costs to identify the structure, as we don't have SIN in India?
It depends on the protein. Sometimes making the protein is very challenging, especially if you're talking about deep protein coupled receptor, which is membrane protein, it's very challenging. If your protein can be made in E. coli, then we can solve a structure right from synthesize the DNA, express in E. coli, express the protein, purify it, express it, solve the structure within like a few weeks, maybe even a month. And the cost again varies, depends where you collect it, but it could be like $5,000, which is around like 4 lakhs. But it can go up. I mean, some cryo-electron microscopy, the costs are coming down, but it could cost millions of dollars for a structure of a calendar.
So next question, sir. Can India afford a synchrotron?
It can, but it's a question of cost benefit, right? I mean, if you can just ship your proteins to another place and then they can collect data and send it back for like few lakhs, would you spend few crores in building a synchrotron? So it's always been discussed from the time I was doing it. Should we build a synchrotron? And now, with the fact that cryo-electron microscopy has caught up with crystallography in terms of the resolution of the structures, I think India probably, I'm sure now it has a bunch of electron microscopes, which also cost a few $1 million. But I'm sure India has a few of those now. And that would be the way to go. I don't think it makes that much sense for us to build a synchrotron. There were talk, Baba Atomic Research Center in Bombay was thinking about building a synchrotron, but finally it never got done. Basically because it didn't make, even the amount of structures we determined in India, it didn't make sense to build a synchrotron.
So next question, sir. At what stage protein ligand docking will happen?
You mean in the process of drug discovery or in so okay, just to explain the question to everybody. Protein ligand docking is that when you don't have a structure of your protein with your small molecule ligand, but you have a structure, apostructure of a protein. In the computer, you can say, hey, the shape of this protein molecule is this. Will my small molecule fit into that protein? Now, there are lots of software which can do that, and some of them work reasonably well, but we never know because the software is not accurate enough docking. So we do docking because it is very cheap. You can do it on the computer. You don't have to express your protein. You don't have to grow crystals. You don't have to collect data. We do docking all the time, but that is more as a supplement to determining structures as opposed to a replacement.
So next question, sir. How does a clinician contribute to the field of drug discovery? What are the avenues available in terms of career options?
Different ways, right? So if you join a pharmaceutical company, then doctors in pharmaceutical companies play multiple different roles. When they have a deep understanding of human pharmacology, which can guide you in saying like, hey, these are the tests we should do in animals. These are the, like, in determining what kind of doses we should give. They have different in designing your clinical trials, what frequency of doses, how many times you should repeat, stop and repeat. So there is that contribution. If you're in an academic institute or a hospital where you have patients, then you can collaborate with the drug discoverers in pharma or in government agencies to do actual clinical trials, which is of course a critical component of approving a potential drug and getting it into the market.
So next question, sir, why is it that new drugs are not discovered in India?
It is a challenging process. Like, I mean, maybe a glimpse of this should show it is really a challenging process. I think some new drugs are discovered. I'm not that familiar with the Indian drug discovery process right now. In the years I've been here, I've not been in that much touch. But I think there are some discovery, which also happens in India. But it is, you can see, it's a very expensive, very long-driven process where the investment, you don't expect a payback immediately, because like I said, if it takes 8 to 10 years to get a drug, that's like over $1 billion you spend at that time, and you don't know whether your drug will succeed in the clinic. So that is a challenging aspect. And there is another aspect of drug discovery where you take an approved drug and see if you can make it better. So the first drug to treat a particular disease we call first in class. But then sometimes you find, like, the cancer drugs, they have resistance which develops in the clinic once you start using it, and then you want to develop a better drug. You can, that is a little easier to do than to develop the first drug, because the first drug you're even trying to validate, is this protein target a good target for drug? There's a lot of that, and patent busting is something you take a drug and see whether you can circumvent the patent or making a change sufficient so that it's not covered by the previous patent. I mean, I don't have a great opinion of such an approach, but it still works in the same efficacy. So that's a complicated question. I don't have the full answer for why India is not more active in drug discovery.
So just continuation to that same question, isn't it not having a synchrotron a handicap for that?
Not really. Like I said, I really don't think having a synchrotron is a big handicap. Like I said, you can ship crystals. There are a number of synchrotrons in the US, in Europe, in Japan, in China, in Australia. They are all happy to collect data. There was a time when getting time to collect data at a synchrotron was a scarcity, and you had to wait in a queue to get into. You had to write a very compelling argument why that is important. But that time is gone. Now all the synchrotrons have enough time, and especially if you are a non-profit or an academic institution, synchrotrons will collect data for free. I don't think you can ship it like FedEx. 3 days later, it's in US. You collect it, and then data is transferred back. I really don't think that's an excuse for not doing drug discovery, that we don't have a synchrotron.
Oh, one more question. Previously, people were told there'll be thousands of molecules to try out before you get one in clinic which works, and that's billions of dollars. With this kind of lab-based, this PDBB, is that becoming less and less, the amount of amount you have to spend before you get one successful molecule? Does that still hold good?
It still holds good. That's the sad part of it comes. If you look at the cost of developing drugs, like 5 years ago or 6 years ago, there was this paper which said it was around $0.7 million. Sorry, for $0.7 billion. So 700 million. But now the cost has gone over $1 billion, $1.5 billion. So even though that you are trying to find new drugs, the cost has gone up. The primary reason, let me explain, is this. For many of the easily treatable diseases, we have drugs, and those drugs, many of them are off patent. So many of those drugs have come off patent.
Can everybody mute themselves?
Pardon me,
No, I think people are speaking.
Yeah. So the cost has really not gone down. It definitely has made things a little bit more efficient. The more we learn about at every step of the process how efficient you can be, that brings the cost down. But the overall cost, just because we are tackling much more challenging diseases, much more difficult to tackle diseases, the cost has gone up. The clinical trials have become more expensive. The molecules you need to find have become more difficult. And the fact is, right now we have a whole bunch of generic drugs out in the market which can treat many of the diseases, right? But of course, there are many treatments, many diseases which we can't treat. And when you have somebody near and dear who's affected by those, you feel the pain. How are we doing for time, Thomas?
I think we are near the end. I think Mohan, any more questions?
There are no further questions are in the chat box.
Okay. So it's my pleasant duty to thank you, Suresh. Dr. Stephen Antonisamy studied in Scudder School, so all people from Vellore can be very proud of him. Thank you very much for taking this time to take this class for us. I think it was very apt for our department, physicians, or anybody else listening, because often it's a mystery how things are made. We do have a subject in pharmacology on all drugs and other things, and a little bit about drug discovery, but this has taken the talk to another level, and I think our understanding also has opened up. I guess as physicians we may wonder how we can be part of the process, but I guess we are at the other end of it, at the clinical trial end of it, or perhaps even some physician who's listening to the talk, who's interested, who wants to identify a new target for a new disease. Maybe he will get involved and sort of listen to this. So I hope you don't mind us sharing this talk on the net with other people so that they can also be inspired. And if anybody would like to get in touch, we can get in touch through Mohan in the medicine department. So thank you very much again, Suresh. It's been a real honor to listen to you. Thank you.
So thanks, Tom. Thanks for inviting me to give the talk. I'm happy to give the talk. I enjoyed it. Thank you.
Okay. I think there's a last question. If you can take
Sure.
this last one. As the
Sir,
cost of getting anything done in India is usually less expensive, wouldn't the cost come down if drugs are developed in India?
For sure, Tom. For sure. Yes. Um, I think that's a very good argument for making drugs in India. Yeah, for sure. And especially you can. There is also the issues of the patent situation, right? The Indian patent situation is different from US. So I don't know. Maybe you can't protect it that much. So the patent gives you a certain amount of years where you can try and recoup the amount you spent in the. Maybe the Indian patent landscape is not that favorable. So that's the tricky bit. You don't want pharma companies to make too much money from the drugs, but you want them to make enough money that they will continue making new drugs, right? If they decide, hey, this is not as profitable, they'll start making sandwiches or burgers. I don't know.
And on that wonderful note, I think we'll take an end to our talk. I think people are asking, what can you talk about next? Is there some other area you'd like to give us a talk on little later on? Something else on drug discovery, a little more in depth in some areas?
So we can talk. We can think about it. We can
Sure,
discuss about it. Yeah.
sure, sure.
I mean, like this was kind of an overview and a little bit of depth on structural biology, but each of those topics we can get into more detail. But just also, I was not sure what the right content was for the audience.
I think this is fantastic. This is fantastic. This is just the right one. Apt for us. Thank you so much.
Thanks. Thank you.
All right. So everybody, thank you all. Thank you to Mohan for arranging this. And everybody have a good day and have a good evening, Suresh. All
Okay.
right.
Bye
Okay,
bye.
everybody. You can listen to this talk on YouTube in probably a week in our department channel. And if you have any topics which you'd like us to take, just please email the department and we'll try to accommodate you. And we'll have another talk by Suresh probably then in 2021. We can decide on what aspect we can get him to talk to us about. So thank you all. Good night.
Thanks, Tabu.
Okay, bye.